Welcome to the Sleepy Brain Facts channel. Tonight, you will embark on a journey into the fascinating world of DNA, those tiny spirals of code that not only shape the natural world, but also make each of us unique. DNA is life expressed in a language so minuscule that it resides at the very core of our cells, yet it encompasses the entire history of life on Earth. It serves as the microscopic mechanism behind growth, memory, healing, and the endless transformations that occur in the living world. In this molecular realm, you will discover the very foundations of creation. Instructions flow quietly through generations, constructed with remarkable precision. DNA weaves forms, builds structures, and harbors mysteries that trace back to the earliest moments of biology. It is a domain where chemistry transforms into memory and where the tiniest details guide the grandest wonders. If you find solace in these tranquil explorations, consider liking, subscribing, or sharing your thoughts below. Your engagement helps others discover this space, one sleepy soul at a time. For now, though, all you need to do is relax, take a deep breath, let your shoulders drop and allow your mind to unwind gently. Let's begin. Imagine this: a single gram of DNA could hold more data than all of human technology combined. Imagine if all the digital output of humanity could be condensed into a physical object. You might envision a tiny speck of genetic material. The essence of life is so compact that it holds vast amounts of information within a space even smaller than a sugar crystal. This remarkable feat is possible because each minuscule unit in the molecule acts like a symbol, and these symbols can be arranged in nearly infinite combinations. Unlike our digital machines, this biological form of storage doesn't rely on electricity or ongoing maintenance. When kept in stable conditions, the sequence can remain readable for far longer than any modern device could last. Scientists have already ventured into the realm of embedding texts, images, and even simple moving scenes into these strands, retrieving them with astonishing accuracy. The concept that living chemistry can serve as a lasting archive suggests a future where libraries merge biology with information science, creating a bridge between the realms of nature and technology. DNA stands as the most sophisticated information system known to us. Life employs a coding system that is both incredibly compact and remarkably powerful. Within every cell, this molecular script directs how bodies grow, repair, and adapt. As you reflect on this, consider the profound implications it holds for our understanding of information and life itself. Wouldn't it be fascinating to explore how these connections might shape our future? Feel free to continue pondering or take a moment to rest with these thoughts. What makes this phenomenon truly extraordinary is not just its precision, but also how it seamlessly integrates shape, timing, and instruction all in one place. It contains long sequences that function like sentences, regions that act as signals, and patterns that dictate the timing of its messages. Rather than merely storing information, it constructs the tiny machines that interpret and execute the very code it holds. The same set of instructions can guide a cell in building the components it needs, organizing them effectively, and adapting to changes in its environment. It operates continuously across countless cells without losing clarity. Throughout this process, the information remains stable enough to be passed down through generations, yet adaptable enough to accommodate new traits as life evolves. Every strand of DNA tells a story encoded in a four-letter alphabet. Along each molecule lies a sequence of repeating symbols that form the most profound biography any organism can possess. At first glance, these symbols may seem simple, but their specific arrangement directs the creation of structures, pigments, enzymes, and everything else that sustains a living body. Some segments dictate the strength of a muscle, while others determine how a cell reacts to variations in temperature or light. In various biological contexts, groups of symbols collaborate to create rhythms, pauses, and signals that guide cells on when to start or stop their activities. Between these regions lie segments that determine the speed at which each message is conveyed. Even the slightest change can influence how a molecule folds or how an organism interacts with its environment. By analyzing these sequences, researchers can trace ancient lines of inheritance and observe how traits have evolved over time. DNA possesses an incredible resilience, surviving in conditions where very little else can endure. Long after soft tissues have decomposed and bones have disintegrated, small fragments of genetic material can remain safeguarded in cold soils, arid caves, and other environments where time unfolds slowly. The structure of the DNA molecule, along with the bonds that maintain its integrity, allows certain parts to resist degradation for remarkable periods. Even when faced with heat, moisture, or natural radiation, some segments remain intact enough for scientists to decode them. These enduring fragments provide insights into the types of plants and animals that once inhabited a region, how populations migrated, and the environmental conditions that shaped them. In some extreme habitats, tiny organisms employ repair mechanisms that stitch their genetic material back together after it has been damaged, enabling them to thrive in conditions that would obliterate most life forms. As you reflect on this, consider the remarkable resilience of life and the stories hidden within these ancient genetic fragments. Each strand of DNA carries a message from a distant past, waiting to be uncovered and understood. This molecular structure is a remarkable feat of architecture. Two long chains twist gracefully around each other, forming a shape that is both beautiful and functional. This spiral design provides the strength needed to endure countless cell divisions while remaining flexible enough to navigate tight spaces. Inside the spiral, pairs of matching symbols align in a specific sequence, creating a stable pattern that can be replicated with astonishing accuracy. The outer layer consists of repeating units that lend support to the entire structure, much like the rails of a ladder. This design is not merely aesthetically pleasing, it enables proteins and enzymes to locate their precise interaction points without disrupting the molecule as a whole. When it's time to replicate the information, the spiral unwinds, allowing each chain to assist in creating its counterpart. This entire structure embodies a perfect harmony of beauty, strength, and precision. DNA can also fold into forms that are far more intricate than the well-known double helix. Within the cell, genetic material transforms into more than just a simple twist. It wraps around small disc-shaped proteins that function like spools, forming beads in a repeating pattern. These beads connect and fold into thicker fibers, which loop and curl into larger structures that fill the nucleus. This intricate folding is meticulously organized. Some segments are tucked away, reserved for instructions that are seldom needed, while others remain accessible for quick retrieval. Special helper proteins serve as anchors, securing certain loops in place and establishing neighborhoods where related instructions converge. This folding pattern shifts based on the type of cell and its immediate needs. For example, a nerve cell arranges its loops differently from a skin cell, allowing each to specialize effectively. By organizing itself in this complex manner, the molecule ensures that the right instructions are available precisely when needed. The instructions within DNA can be interpreted in various overlapping ways. A single region may convey multiple messages depending on how the cell initiates its reading. At times, a change in the starting point can yield an entirely different outcome. In other instances, the cell takes the initial transcript and rearranges certain segments, generating new results from the same sequence. As you reflect on this intricate process, consider how the remarkable organization within cells plays a crucial role in their function and specialization. It's a fascinating dance of molecular mechanisms that invites you to explore further, or simply to pause and appreciate the complexity of life at its most fundamental level. Within the intricate world of genetics, hidden signals reside within longer stretches of the genome. These signals play a crucial role in determining when specific regions become active, even though they don't directly contribute to the proteins produced. Some sequences govern the pace at which genetic messages are read, while others affect how the final molecule folds. This layered approach enables the genome to maximize space while supporting a diverse array of functions. Rather than assigning each instruction to a distinct area, life artfully intertwines multiple roles within the same segment of code, achieving remarkable efficiency and complexity. DNA exhibits an incredible ability to repair itself. Cells are in a constant state of vigilance, monitoring their genetic material for any irregularities, bumps, bends, or misplaced units that might indicate damage. When abnormalities arise, specialized enzymes spring into action. Some enzymes pry apart the affected region and excise the flawed segment, while others meticulously reconstruct the missing part, using the undamaged strand as a template. Additional enzymes work to reconnect the backbone, restoring the molecule to its complete form. During the process of replication, proofreading systems diligently check the new copy to ensure that no errors slip through. When breaks occur on both sides of a strand, cells have two options. They can either fuse the ends back together or reconstruct the missing segment by copying from an intact region. This remarkable orchestration of repair and regulation showcases the genius of genetic design. These systems operate continuously, making swift decisions to address various problems. This ongoing vigilance maintains the stability of the genome over extended periods, despite exposure to numerous stressors. Without this repair work, cells would quickly accumulate harmful changes that could disrupt their fundamental functions. Evolution is a relentless force, constantly rewriting DNA. Over generations, subtle adjustments emerge in the sequences passed from parents to offspring. Some of these changes occur when a copying error slips through the proofreading mechanisms, while others stem from environmental influences that gently nudge the molecule into a slightly different shape. Most of these alterations have minimal impact, but a select few can change how a protein functions or adjust the activity of a regulatory region. When a change confers an advantage in a specific environment, it spreads more readily. Over time, these shifts accumulate, leading to new traits, new behaviors, and occasionally new species. Certain segments of the genome can also be duplicated, creating extra copies that can explore new roles, while the original continues its vital function. Other segments may shift positions, change orientation, or become rearranged in unexpected ways. All these transformations turn the genome into a living record of adaptation, molded by countless forces over vast stretches of time. As you reflect on this intricate process, consider how remarkable it is that life continually evolves, adapting in response to its ever-changing environment. Would you like to delve deeper into this fascinating journey of life, or perhaps take a moment to pause and absorb this knowledge? Your DNA holds whispers from ancient relatives you may never have met, Within your genetic makeup lie fragments that trace back to populations once isolated in distant lands. As communities migrated and encountered one another, bits of their genetic legacy flowed into new groups, carried forward through the ages. Some of these fragments signify adaptations to local climates or food sources, while others preserve traits once deemed valuable but now less apparent. Each segment serves as a gentle reminder of our shared human history. By examining these patterns among individuals, researchers can piece together migrations, cultural transformations, and interactions that might otherwise fade from memory. This creates a molecular map of ancestry that extends far beyond the reach of written records, showcasing the deep connections among human populations. Even when separated by vast oceans or towering mountains, DNA can traverse great distances within your genome. Certain stretches of genetic material act like tiny travelers, not remaining fixed in one place, but instead journeying to new locations within your genetic landscape. As you reflect on this intricate web of connections, consider how your own story intertwines with the stories of those who came before you. These wandering segments navigate through natural cellular processes, sometimes venturing into uncharted territories. Their movement is far from chaotic. Many of them utilize specific enzymes that assist in their departure from one location and their establishment in another. Upon arrival, they can influence the activity of nearby genetic instructions. They might awaken a previously silent region or disrupt one that was once highly active. Additionally, they can forge new combinations of sequences by transporting small fragments of surrounding material with them. While their movements may seem risky, they have been a vital source of new adaptations throughout evolutionary history. Over vast stretches of time, these traveling sequences have shaped genomes, introducing diversity and fresh potential that simple copying could never achieve. Their roaming behavior transforms the genome into a dynamic landscape, constantly shifting in subtle and unexpected ways. Within this landscape, genetic switches play a crucial role. These small regulatory regions act like switches, controlling thousands of biological events throughout the genome by turning instructions on or off based on the cell's needs. Interestingly, these switches do not encode structures or enzymes, but they are essential for the intricate dance of life. Instead, you discover that certain messages are read at specific times, with varying intensity, shaping how a developing organism responds to internal and external signals. Some switches activate only under particular conditions, like a sudden increase in temperature or a shift in nutrient availability. Others operate in a rhythmic manner, aligning with daily biological cycles. A single switch can influence many seemingly unrelated processes by controlling a region that branches into multiple pathways. As several switches interact, the complexity grows significantly, enabling the intricate patterns of growth and specialization found in all forms of life. Even minor changes in these regulatory zones can lead to far-reaching effects, resulting in variations in shape, color, behavior, or physiology. This serves as a profound reminder that the genome is more than just a list of components. It is a finely tuned system of timing and control. Silent regions of DNA harbor secrets that shape your existence. Among the areas that actively guide the production of molecules, there are long stretches that may seem quiet at first glance. Although these regions do not directly code for proteins, they play crucial roles in directing how the genome operates. Imagine a world where tiny elements play significant roles in the grand design of life. Some regions of DNA act as landing pads for regulatory proteins, guiding whether nearby sequences become active. Others provide essential structural support, helping the entire molecule fold into specific patterns that determine which sections are accessible. You might find it intriguing that some silent stretches serve as spacers, keeping critical regions at the right distance from one another. These regions can also carry signals that influence how quickly a message is read or how a cell responds to developmental cues. Once considered unimportant, ongoing research reveals that these quiet areas significantly impact traits from immune responses to physical features. Even though they may not make a lot of noise, their presence subtly shapes the flow of information in countless ways. DNA is a marvel, forming loops and tangles to fit inside a tiny cell. A single molecule of genetic material is far longer than the cell that houses it, yet it fits comfortably due to a remarkable folding system. The molecule bends, curls, and loops into compact shapes, protecting it from damage and preventing tangles. Some loops bring distant regions close together to coordinate activity, while others separate conflicting areas to maintain order. As you reflect on this intricate dance within your cells, consider how even the quietest parts contribute to the symphony of life. Would you like to explore further, or perhaps take a moment to rest? Special helper proteins play a crucial role in guiding the folding process of DNA. They hold certain sections together while loosening others as needed. The structure of DNA shifts depending on the cell's activities. When a specific region needs to be accessed, that loop relaxes and opens up. Conversely, when it is not in use, it tightens and becomes more compact. Each type of cell organizes these loops in its own way, creating specialized patterns that enable it to fulfill its unique functions. This intricate arrangement allows vast amounts of information to operate efficiently within an incredibly small space. Interestingly, some DNA exists outside the nucleus. While the majority of genetic material is housed within the central compartment of a cell, a smaller fraction resides elsewhere. Tiny structures responsible for energy production contain separate circular molecules that carry their own set of instructions. These instructions are essential for maintaining the machinery that powers cellular processes. This external genetic material is inherited through a different pathway than the sequences found in the nucleus, offering a parallel thread of ancestry that extends through many generations. Because it operates independently, this genetic material can evolve at a different pace, leading to variations that may not manifest in the main genome. Moreover, these independent molecules interact with nuclear genes, forming a cooperative system that ensures the cell functions smoothly. Consider how this intricate interplay supports life at its most fundamental level. You might find it fascinating to reflect on the complexity and beauty of these processes. If you're curious to explore further, feel free to continue your journey or take a moment to rest and absorb this information. Their existence illustrates how genetic information can be spread across various locations within a single organism. Each segment plays a crucial role in its own unique way. Remarkably, DNA can be transformed into sound and music. Researchers have discovered innovative methods to convert genetic sequences into audible patterns by assigning distinct tones to each symbol within the molecule. When these tones are played, they create melodies that reflect the intricate structure of the DNA. While these sounds are artistic interpretations, they unveil patterns that might be missed when observing the sequences solely as text. Repeated stretches resonate with rhythm, rare combinations produce unexpected notes, and lengthy pauses introduce moments of stillness. Composers and scientists have come together to delve into these patterns, turning biological information into a fresh sensory experience. This transformation doesn't alter the scientific essence. Instead, it offers a new lens through which to view the complexity and order embedded in the genome. It showcases how information from living systems can transcend into realms like music and design, igniting curiosity and paving the way for novel understandings of life's hidden architecture. You carry ancient viral DNA within your cells, a reminder of the intricate connections that bind us to the history of life itself. Long ago, certain viruses embedded their genetic material into the genomes of the organisms they infected. Rather than being eliminated, many of these viral sequences became permanent fixtures, passed down from parent to offspring through countless generations. Over time, most of these sequences lost their ability to function as active viruses, yet their fragments linger throughout the genome. In some cases, these sequences evolved new functions. They can influence nearby regions, engage in regulatory processes, or serve as raw material for new adaptations. Their presence provides a fascinating glimpse into ancient interactions between species and the viruses that once circulated among them. By examining these remnants, scientists can trace historical outbreaks, uncover how viral elements have influenced evolution, and understand how genomes protect themselves from new infections. These embedded sequences serve as records of long-ago battles preserved within modern cells. DNA creates proteins through an elegant molecular assembly line. Within each cell, a sophisticated system reads instructions and transforms them into functional molecules. This intricate process begins when a segment of the genome is copied into a temporary form that can be transported. As you consider this remarkable journey of genetic material, you might reflect on the resilience and adaptability of life itself. The stories encoded in our DNA remind us of the interconnectedness of all living things. Would you like to explore more about how these ancient interactions shape our understanding of evolution today? Imagine a temporary copy navigating a tiny structure, meticulously coordinating each component to position every building block just right. As this growing chain unfolds, it forms intricate shapes that define its purpose. Each movement is guided by specific interactions between the temporary copy and the building blocks it assembles. The entire system operates with astonishing precision, generating countless proteins that handle essential tasks, from transporting oxygen to breaking down nutrients. This assembly line works tirelessly, adapting to the cell's needs and responding to environmental signals. It transforms simple sequences of symbols into the diverse machinery that sustains life at every moment. In this microscopic realm, microorganisms engage in a remarkable exchange of DNA, acting as tiny biological messengers. Some organisms share genetic material with surprising ease, extending small connections to transfer segments of DNA directly to a neighbor, or releasing fragments into the environment for nearby cells to absorb. Some even harness viral light particles to convey information between individuals. This dynamic exchange enables them to swiftly acquire new abilities, such as resistance to toxins or the capacity to utilize unfamiliar food sources. As you reflect on this intricate dance of life, consider the profound interconnectedness that sustains existence at such a fundamental level. These transfers do not rely on traditional reproduction, allowing them to spread swiftly through communities, much faster than inherited changes. This rapid sharing fosters dynamic populations that adapt in real time to changing conditions. It also highlights the remarkable flexibility of genetic material, which can move between organisms and reshape entire ecosystems. DNA has the capacity to record memories within living cells. Scientists have created techniques that enable cells to store traces of past events directly in their genomes. By linking specific processes to controlled modifications in designated regions, a cell can leave a molecular note indicating that something significant occurred. Over time, these changes accumulate, forming a sequence that mirrors the unique experiences of the cell. For instance, a sudden shift in the environment might trigger a minor alteration in one area, while a different event leaves its mark elsewhere. Although these edits do not operate like conscious memory, they create a record that researchers can later interpret. This innovative approach opens new avenues for tracking how cells develop, respond to various signals, and interact with their surroundings. It transforms the genome into an active logbook, capturing the history of individual cells in a way that traditional tools simply cannot achieve. Certain organisms possess the remarkable ability to rearrange their DNA throughout their lives. For some species, genetic material is not a fixed entity from birth. Instead, it undergoes intentional restructuring as the organism matures. Take, for instance, some single-celled creatures that fragment their genomes, discarding large segments while stitching together the remaining pieces into a configuration that better suits their adult form. Other organisms modify specific areas of their DNA to generate new combinations of instructions, aiding their adaptation to changing environments. This process is intricate and relies on specialized enzymes that identify particular patterns, cutting or joining segments at exact locations. The outcome is a genome that evolves within a single lifetime, adjusting to new challenges rather than relying solely on inherited mutations over many generations. This internal editing process can shape feeding strategies, defense mechanisms, and developmental transitions. It highlights that genetic material is not merely a static blueprint. In certain forms of life, it acts more like a flexible script, one that can be rewritten to meet immediate needs. Nature employs molecular scissors to edit DNA. Within living cells, proteins function as tiny tools, executing precise cuts in specific regions of the genome with astonishing accuracy. Imagine a world where natural cutters identify specific patterns within molecules, slicing them precisely to create opportunities for repair or alteration. The cell then steps in, utilizing various pathways to mend these breaks. Some pathways simply connect the ends, while others introduce new sequences or replicate information from similar areas. Viruses, bacteria, plants, and animals all depend on these natural editing systems for self-defense, adaptation, and the maintenance of healthy genomes. Certain microbes harness these cutters to eliminate unwelcome genetic material, while plants and animals use them to excise potentially harmful sections. Over extensive stretches of evolutionary time, these editing events play a crucial role in developing new traits and structural changes. The existence of such precise natural tools highlights how actively genomes are shaped by intrinsic mechanisms that manage, refine, and at times reinvent their own sequences. A single change in DNA has the power to transform an entire species. In rare instances, one small adjustment within a lengthy chain of symbols can redirect the course of evolution. This modification may affect a protein responsible for guiding early development, leading to changes in limb arrangement, color patterns, or the shape of sensory organs. Consider the profound impact of these natural processes on the diversity of life around us. Each adjustment carries the potential for transformation, revealing the intricate dance of evolution that shapes our world. Imagine a world where tiny adjustments in our genes shape the very fabric of life. You might not realize it, but even the smallest changes in a regulatory region can dictate how often a gene is activated or under what circumstances. These subtle differences, seemingly insignificant at the molecular level, can lead to remarkable variations in anatomy and behavior. Over generations, when these differences confer advantages in survival or reproduction, they may spread widely throughout a population. In time, they can even give rise to new branches on the vast tree of life. While most changes are minor, history has shown that occasional significant alterations can create ripples that influence entire ecosystems and reshape lineages. This delicate sensitivity highlights how deeply interconnected biological diversity is with the intricate details of the genome. Within DNA lies a treasure trove of instructions for constructing millions of different molecules. Each organism possesses the potential to develop an astonishing variety of molecular tools, all derived from the information encoded in its genome. Some of these instructions guide the creation of enzymes that accelerate chemical reactions, while others focus on structural components that provide tissues with strength, flexibility, or color. Moreover, many sequences generate small regulatory molecules that enable cells to communicate and coordinate their functions. By mixing and matching various building blocks in countless configurations, cells craft molecules specifically designed for distinct tasks. This intricate process underscores the beauty and complexity of life itself. Imagine a world where living systems thrive, adapting to an incredible variety of challenges, from processing nutrients to sensing changes in the environment. These systems exhibit a remarkable diversity. This diversity is not fixed, it evolves. Over generations, new molecular varieties emerge as sequences shift and expand, providing the foundation for further adaptations. Consider how this ability to produce a wide range of molecules enables life to flourish in diverse habitats, from the depths of ocean vents to the lushness of tropical forests and the stark beauty of frozen tundras. The structure of DNA plays a crucial role in how life unfolds. While the sequence of symbols along the DNA strand is essential, the physical shape the strand takes also influences how instructions are executed. In certain areas, the molecule bends sharply, creating pockets that attract specific proteins. Other sections twist into tighter spirals, becoming less accessible unless the cell unwinds them intentionally. Some regions form small loops, bringing distant parts of the molecule together in ways that would be impossible if it remained linear. These structural variations are key. They determine which genes are active, how quickly messages are interpreted, and how the genome responds to internal signals. As you reflect on these intricate processes, consider the beauty of life's adaptability and the complexity woven into every strand of DNA. Would you like to continue exploring this fascinating world, or perhaps take a moment to pause and absorb what you've just learned? Different cell types arrange their DNA in unique ways, which means that the same sequence can lead to different outcomes based on the environment in which it folds. This dynamic shaping process allows cells to exert more control over their genetic material, adding complexity and adaptability to living systems. Interestingly, some species possess entire extra genomes hidden within their cells. A surprising number of organisms carry more than one complete set of genetic instructions. In addition to their primary nuclear genome, these organisms often hold additional collections of sequences stored in separate cellular compartments. These extra genomes play vital roles, supporting specialized functions that the main genome alone cannot perform efficiently. For example, certain plant species contain independent sets of instructions that govern photosynthesis, while some protists maintain multiple genomic systems to manage different stages of their life cycle. These additional genomes often have their own inheritance patterns, evolving at distinct rates and accumulating unique combinations of traits. Their presence can significantly enhance survival by providing the organism with a broader toolkit to draw upon. This layered organization illustrates that genetic information operates on multiple levels simultaneously, with each genome contributing to a larger network of interactions that support growth, energy production, and adaptation to the environment. Your genome holds echoes of ancient genes, remnants of a time long past. Throughout the vast timeline of life, sections of your DNA have been copied and recopied. Some of these duplicates remain active, while others fade into obscurity. Even the ones that are no longer in use can be detected as faint traces among more recent sequences. These copies serve as a record of past expansions, where a gene duplicated and ventured into new functions, all while the original continued its established role. Some duplicates evolved into entirely new tools, shaping significant transitions in evolution. They contributed to the development of new metabolic pathways or unique anatomical structures. Others have drifted into near silence, leaving behind only subtle hints of their former activity. By examining these remnants, researchers can trace the history of genes that once played vital roles in ancient organisms, but have since been replaced or transformed. They reveal a profound truth. Innovation often arises not from the creation of entirely new sequences, but from the replication of what already exists, allowing variation to accumulate over time. Your DNA creates molecular machines that assemble every part of you within each cell. These instructions are transformed into intricate structures that operate with remarkable precision. As you reflect on this complex tapestry of life, consider the journey of your own genetic story and the countless generations that have shaped who you are today. Feel free to pause and absorb this wonder, or continue exploring. The choice is yours. Cells are remarkable entities, crafting tiny machines designed to manage intricate tasks such as moving nutrients, repairing damage, and generating energy. Each machine emerges from a series of steps that connect simple building blocks into long chains, which then bend and twist into precise shapes tailored for their specific roles. Some of these machines capture light, while others transport ions, and many join forces to form larger networks that facilitate communication across various tissues. The precision with which these structures assemble is truly astonishing, particularly in the bustling environment of the cell. Even minor adjustments in shape can significantly alter their capabilities, and slight variations in the underlying instructions can lead to entirely new functions. Together, these machines carry out essential work that enables living organisms to grow, sense their surroundings, and adapt to changes. This process illustrates that the information contained within the genome is not merely descriptive, it is profoundly constructive. Epigenetic marks play a crucial role in this dynamic. They can modify gene activity without altering the DNA itself. Cells employ chemical tags to designate certain regions of the genome, influencing how actively each section is utilized. These tags attach to specific sites, determining whether nearby instructions remain accessible and ready for action, or become tightly packed and subdued. As you reflect on this intricate world within cells, consider how these processes connect to the broader tapestry of life. Would you like to explore more about the fascinating interplay of genetics and cellular function? As you delve into the fascinating world of cellular development, you discover how the placement of certain marks shifts, influencing the identity of cells as they specialize. These marks are responsive, adapting to various signals such as nutrient availability, stress, and environmental conditions. Importantly, These modifications don't change the fundamental sequence of DNA. Instead, they function like adjustable settings, fine-tuning the behavior of genes. Some of these marks remain stable through cell divisions, allowing daughter cells to inherit the same patterns. Others are temporary, fading away once the triggering event concludes. This intricate system offers flexibility, enabling organisms to react swiftly without needing to rewrite their genetic code. It introduces an additional layer to gene regulation, allowing the genome to respond differently as conditions change. Imagine DNA as its own chemical logic system. Within the genome, certain regions interact in ways that resemble simple decision-making processes. Some sequences activate only when multiple conditions align, while others suppress activity unless specific molecules are present. These interactions stem from the shapes the strands take, the availability of helper proteins, and the presence of chemical tags. Together, they form pathways that respond to signals in a reliable manner. As you contemplate these complex mechanisms, consider how they shape life itself. Wouldn't you agree that understanding this intricate dance of biology opens up a world of possibilities? Feel free to explore further or take a moment to reflect on this remarkable process. Certain regions function like gates, opening only when the right combination of factors comes together. Others act as checkpoints, ensuring that instructions are not used too soon. This intricate network enables cells to coordinate their growth, adjust their behavior, and maintain stability. Instead of depending on a central control system, the genome relies on local signals to generate organized responses. The outcome is a self-regulating system where information and structure merge to guide complex patterns of activity. Inside every living cell, tiny molecular motors glide along strands of genetic material, reading the DNA sequence as they navigate. These motors harness bursts of chemical energy, propelling themselves forward, step by step, while keeping track of their position, even amidst the bustling interior of the cell. As they advance, they gently pull apart the strands to reveal the symbols that need interpretation. They create a temporary copy of that specific region, allowing the instructions to be utilized elsewhere. Different motors have specialized roles in this process. Some are dedicated to copying long stretches of DNA during cell division, while others focus on shorter segments to manage daily cellular functions. This division of tasks ensures that the cell operates smoothly and efficiently, adapting to its needs as they arise. The speed, accuracy, and endurance of these chemical engines are truly remarkable. They navigate intricate twists, loops, and shifting folds within genetic material. Without them, cells would struggle to access the vital information necessary for life. Their activity reveals how dynamic the genome becomes once life begins to harness it. While DNA is often depicted as a simple spiral, certain regions can twist into fascinating shapes. In areas where symbols repeat frequently, the strands can form tight knots, stabilized by unique chemical interactions. In other regions, 4 separate strands may converge to create a compact 4-stranded block, proving to be sturdier than the usual double chain. These shapes can appear and disappear based on the cell's internal environment, the activity level in specific areas, and the presence of particular helper proteins. When these structures form, they can significantly influence how easily a motor reads that section, how frequently a gene is expressed, or how tightly the strand folds. Some shapes contribute to stability, while others may serve as road signs, slowing down the reading process until the cell is prepared. Isn't it fascinating how life continually adapts and organizes itself at such a microscopic level? You discover that the genome holds far greater structural diversity than the familiar double helix implies. DNA stores energy in ways that scientists are still striving to comprehend. Within this twisted strand of genetic material, subtle physical forces gather, shift, and release in patterns that remain only partially understood. As the strand twists, bends, or tightens, energy is captured in these movements. This energy plays a crucial role in how the molecule interacts with enzymes, how quickly regions unwind, and how tightly certain loops stay coiled. Researchers investigating these properties find that even minor changes in shape can affect the distribution of tension along the strand. This tension determines which areas become more accessible and which remain sealed. The notion that a molecule which contains instructions also harbors physical energy that influences its own behavior is captivating. It implies that the genome is not merely a passive script, it is an active structure that employs physics to govern itself. As scientists delve deeper into these forces, they unveil new insights into how mechanical properties impact biology, revealing layers of control that are often overlooked outside of specialized research. You find yourself drawn into this fascinating exploration, eager to learn more about the intricate dance between structure and function within the genome. Your genome holds echoes of ancient extinctions. Hidden within your genetic makeup lie faint traces of lineages that have long since disappeared. Once populations thrived, expanded, and ultimately vanished due to changes in climate, migrations, and natural events. Before these groups faded from existence, they passed fragments of their genetic identity to the ancestors of modern humans. Over the ages, these fragments blended with others, creating a rich tapestry of genetic diversity. Though many of these remnants serve no clear purpose today, they remain as quiet markers of forgotten communities. By examining these signals across different individuals, scientists can uncover population patterns that lack written records. Some regions reveal groups that adapted to environmental shifts, while others trace histories of communities that intermingled and then faded away. These tiny echoes are rarer than well-known ancestral markers and require meticulous analysis to uncover. They offer a glimpse into ancient worlds shaped by forces that left few visible traces. DNA communicates through swift chemical signals, with countless messages racing between molecules inside your cells. Chemical changes signal activity within a region of a molecule, allowing proteins nearby to sense these shifts almost immediately. When one part of the molecule opens up for reading, it triggers a response in other proteins. They adjust their behavior based on this new information, enabling cells to coordinate complex responses in mere moments. Whether nutrients become available, stress levels rise, or a developmental transition occurs, the genome can react in a highly organized manner through these rapid exchanges. Although these signals aren't words or symbols, they convey vital information that determines how the genome is utilized. Their swift nature allows even the simplest cells to adapt quickly to changing conditions. This creates a communication system intricately woven into the very fabric of life. Interestingly, some life forms employ alternative genetic codes. While most organisms follow a universal set of rules for interpreting sequences, certain species have developed unique systems for reading their genetic instructions. In these cases, symbols that typically represent one building block may signify another. These alternative rules often emerge in unusual environments or in tiny organisms that have adapted to very specific lifestyles. Imagine a world where genetic machinery has evolved over countless years, adapting and shifting to create new systems that still function seamlessly. This flexibility in the genetic code reveals a remarkable truth. life is capable of reinterpreting its symbols while maintaining stability and functionality. These alternative systems provide scientists with insights into how early genetic rules might have emerged and how living organisms can explore new configurations without losing order. What if you considered the idea that the basic logic of biology is far more varied than we once thought? DNA holds molecular clues to the earliest living cells on Earth. Strikingly, some of the oldest characteristics of genetic material are found across all known life forms. This suggests that these features were present in the planet's first organisms, guiding essential tasks such as reading genetic instructions, copying genomes, and managing energy. As many species continue to utilize these foundational elements, researchers propose that these features originated in the first simple cells that emerged billions of years ago. By comparing modern genomes, scientists can uncover details about how early life stored information, maintained stability, and adapted to primitive environments. Certain repeated patterns and conserved regions serve as molecular fossils, offering a glimpse into our planet's distant past. Take a moment to reflect on this journey of life and the intricate dance of genetics that has shaped our world. Would you like to delve deeper into this fascinating subject, or perhaps take a moment to rest? You find traces of the earliest biological solutions that enabled life to endure. By studying these clues, you uncover how complex organisms emerged from simple beginnings, offering a rare glimpse into life's initial steps. Each day, your DNA undergoes tiny changes. Within every cell, the genome experiences slight alterations due to natural processes. Everyday activities, like copying molecules or exposure to common chemicals, can shift a symbol into a new form. While cells efficiently correct most of these changes, a few remain as harmless variations. Throughout your lifetime, these small differences accumulate in various cells across your body. This means that the genetic material in one part of your body may differ slightly from that in another. Most of these changes are neutral, yet they contribute to the subtle variations that exist even within a single individual. This ongoing adjustment reveals just how active and dynamic your genome truly is. It is not a static entity. Rather, it is a constantly maintained and ever-changing system. Consider the DNA found in the dark depths of the ocean. It functions differently than the DNA found on land. Organisms living far beneath the surface contend with unique pressures, temperatures, and chemical environments that are unlike anything on land. Imagine a world where life thrives in conditions that would challenge most other organisms. Here, genetic material functions under extreme circumstances, relying on unique adaptations that keep it stable and ensure accurate replication. Deep-sea creatures, for instance, possess specialized proteins that tightly bind to their genetic strands, safeguarding them against the harsh environment. Some organisms employ specific chemical modifications to prevent their DNA from unraveling, showcasing the resilience of life. Their genomes are tailored to meet the demands of cold, high-pressure surroundings, guiding the production of molecules that are essential for survival. These remarkable adaptations remind us that the genetic code is not uniform across all species. Instead, it responds dynamically to environmental challenges, allowing life to flourish in seemingly inhospitable places. Within this genetic code, there are hidden mathematical patterns. As researchers delve into the arrangements of symbols, they uncover relationships that echo mathematical structures. This intricate coding groups certain building blocks in ways that minimize the risk of harmful errors, creating patterns that enhance resilience. Additionally, scientists observe how similar genetic sequences behave across various species, revealing even more about the interconnectedness of life. As you reflect on these discoveries, consider the wonders of adaptation and the complexity of life itself. Take a moment to appreciate the resilience found in the depths of our oceans. You might find yourself curious about what other secrets the natural world holds. Patterns within genetic sequences may not be immediately apparent through a casual glance. However, when large sets of data are examined, these relationships come to light. Some scientists propose that these connections reflect fundamental principles that guided the early stages of life, while others suggest they emerged through natural selection, driven by the necessity for efficiency and reliability. Regardless of their origins, these patterns indicate that the genetic code is not merely a random assortment of assignments. Instead, it has an internal structure that fosters stability while enabling evolution to explore new avenues. Within every genome, certain genes remain dormant for most of an organism's life, waiting for specific circumstances to trigger their activation. Some genes may only activate under extreme conditions. For instance, some turn on when temperatures exceed a certain threshold, equipping cells to defend themselves against heat stress. Others respond to low oxygen levels or sudden shifts in salinity. There are genes that activate during drought, those that respond when nutrients are scarce, and even genes that light up in the presence of certain toxins. This silence during typical conditions does not equate to inactivity. Instead, it represents a state of readiness. These genes hold strategies for rare events that may occur only a handful of times throughout an organism's lifetime. Embrace the complexity and wonder of these genetic narratives as they unfold, revealing the intricate dance of life itself. When the mechanisms finally activate, they play a crucial role in stabilizing the internal environment. They support metabolic pathways and trigger emergency responses, ensuring the organism's survival. This reveals that genomes are not just designed for daily functions. They are also equipped to handle unexpected challenges that require swift adaptation. DNA can twist into supercoils, a protective measure against damage. Inside the cell, long strands of genetic material need to remain compact, organized, and safe. One effective way cells achieve this is by twisting parts of the molecule into tight spirals, known as supercoils. These spirals function like coiled ropes, storing tension that maintains stability and prevents tangling or stretching during normal activities. When enzymes need to access or replicate a specific region, they adjust the tightness of these coils, making the area accessible. Once the task is complete, the coils tighten again, safeguarding the section from potential harm. This continuous adjustment is facilitated by specialized proteins that add or remove twists, ensuring the right amount of tension is maintained. Supercoiling also serves to separate quiet regions from those that are actively engaged, establishing a natural boundary system within the genome. Imagine a world where plants possess remarkable abilities to adapt and survive in the most challenging environments. This protective strategy enables them to function amidst mechanical stress without suffering damage. Within their DNA lies a treasure trove of surprising genetic adaptations. Certain plants carry unique sequences that empower them to flourish in conditions that would be fatal to most species. In soils laden with salt, for instance, some plants utilize genes that effectively expel excess salt from their cells. Meanwhile, in the harshness of deserts, others possess the capability to produce specialized sugars, which help retain moisture and stabilize their tissues against the sweltering heat. In nutrient-poor regions, some plants have evolved adaptations that allow them to break down minerals that are largely inaccessible to other organisms. There are even species that have devised innovative methods for capturing nitrogen, diverging from the well-known symbiotic partnerships typically seen in root systems. These remarkable adaptations have been etched into their genomes over countless generations, shaped by the pressures of their environments and the process of natural selection. Each unique sequence offers a solution to specific ecological challenges. By studying these extraordinary plants, scientists uncover the secrets of survival and resilience. This research not only enhances our understanding of life, but also holds the potential to inspire new agricultural strategies, helping crops thrive in our ever-changing climate. So, take a moment to appreciate the wonders of nature and the lessons it imparts. Would you like to explore more about these incredible adaptations? DNA engages in a complex interplay with water molecules, creating fascinating patterns. While we often think of genetic material in terms of symbols and sequences, much of its behavior is influenced by its relationship with the surrounding water. Each twist and curve of the DNA molecule attracts specific arrangements of water molecules that cling to its surface. As the genome folds, these patterns shift, forming layers of hydration that impact both flexibility and stability. Water molecules can slip into grooves along the DNA strand, aiding proteins in recognizing certain regions and guiding how easily sections can unwind. When the environment becomes more crowded or the concentration of salts fluctuates, these hydration patterns rearrange, affecting the overall shape of the molecule. This delicate interaction between DNA and water influences everything from how genetic instructions are accessed to how the genome adapts to temperature changes. Ultimately, this reveals that DNA does not exist in isolation. Instead, its structure is deeply connected to its immediate surroundings. Your own DNA carries traces of the environments your ancestors endured. Within your genome are segments shaped by the climates and challenges faced by those who came before you. Some sequences have evolved to support life in colder regions, assisting the body in regulating heat or managing energy storage. You notice how adaptations to intense sunlight can shape pigmentation and influence responses to ultraviolet exposure. Certain regions are tied to diets rich in specific plants, grains, or marine foods, all of which affect how your body processes nutrients. When groups migrate into new environments, subtle shifts occur in their genomes. Individuals with beneficial traits are more likely to pass those traits on to future generations. These inherited changes do not define your behavior or identity. Rather, they illustrate the historical interactions between biology and the environment. Today, they serve as molecular footprints, revealing the journeys of those who came before you as they navigated new landscapes and faced unfamiliar challenges. Within the genome, there are vast regions that remain a mystery, even after decades of research. Many sections do not encode proteins or function like well-known regulatory zones. Some may influence how the molecule folds, while others might interact with yet-to-be-identified proteins. Interestingly, certain areas are remarkably conserved across different species, hinting that they perform vital functions, even if their exact roles remain elusive. As you reflect on these intricate connections, consider the depth of history embedded within your own genetic makeup. Would you like to explore further, or take a moment to ponder this fascinating journey? As you explore the fascinating world of genetics, you notice that the roles of various genetic regions differ remarkably from one organism to another. Researchers, utilizing advanced technologies to observe genetic material in motion, are beginning to uncover the potential of these regions to facilitate long-range interactions or stabilize essential sections during the early stages of development. The importance of these areas becomes evident as their removal often leads to subtle yet significant disruptions. These enigmatic regions serve as a reminder that the genome still harbors mysteries even within its quieter spaces. Within every living cell, DNA acts like a molecular clock. Certain segments of genetic material contribute to intricate internal timing systems that help organisms maintain their daily rhythms. These segments generate molecules that rise and fall in repeating cycles, influencing sleep patterns, feeding behaviors, temperature regulation, and numerous other processes. The instructions for these timing systems are embedded directly in the genome, and their patterns are astonishingly precise. When one aspect of the cycle increases, another decreases, creating a self-resetting feedback loop. While external factors like light, temperature, and nutrients can influence the pace, the fundamental rhythm is generated from within. As you reflect on this intricate dance of genetics, consider how these hidden mechanisms shape life as we know it. Would you like to delve deeper into this captivating subject, or take a moment to pause and absorb the wonders of the genome? Imagine a world where different tissues operate on their own unique timing loops, yet all work in harmony through signals that synchronize the entire organism. These molecular clocks facilitate a proactive approach, allowing cells to prepare for regular environmental changes. Instead of merely reacting, they adjust their metabolism and activity in anticipation of what lies ahead. This intricate organization underscores how deeply biological timing is embedded in genetic instruction. Consider the resilience of certain bacteria. Some possess genomes capable of rewriting themselves when faced with threats. In hostile environments, these bacteria respond to danger by modifying their own genetic material. When a harmful virus attempts to invade, they identify fragments of the viral sequence and integrate a copy into their genome. Should the same virus return, these bacteria utilize the stored fragment to activate their defensive mechanisms, effectively neutralizing the intruder. This built-in system functions like an adaptive memory, chronicling past encounters and preparing for future threats. As new viral challenges emerge, the genome becomes increasingly complex, accumulating records of previous conflicts. This dynamic strategy equips bacteria with remarkable resilience. They do not merely defend themselves, they evolve their defenses in real time, reshaping their genomes according to the dangers they encounter. Imagine a world where even the simplest organisms adapt their methods of information storage to meet the challenges of a rapidly changing environment. DNA serves as a powerful tool for reconstructing ecosystems that have long since vanished. Scientists extract genetic fragments from soil, ice, sediments, or ancient remains, piecing together the vibrant communities that once thrived in specific locations. These fragments come from a diverse array of life plants, animals, fungi, and microbes, each leaving behind traces of their genetic material as they interacted with their surroundings. Remarkably, even in the absence of bones or fossils, these molecular remnants endure, revealing insights about which species coexisted, which predators hunted their prey, and how the landscape transformed over millennia. By sequencing these fragments, researchers can create rich snapshots of ancient forests, coastlines, or tundras that existed thousands of years ago. This innovative approach allows scientists to delve into ecosystems shaped by climates vastly different from our own, providing valuable clues about how species adapted to fluctuating temperatures, rising sea levels, or advancing ice sheets. Each recovered sequence weaves another thread into the intricate tapestry of ancient life. Your mitochondrial DNA tells a unique story, distinct from your nuclear DNA. This fascinating interplay of genetics opens a window into the past, inviting you to explore the mysteries of life that once flourished on our planet. Within each cell, there exists a remarkable structure known as mitochondria, which plays a crucial role in energy production. These tiny powerhouses carry their own genetic material, referred to as mitochondrial DNA. Interestingly, this mitochondrial genome is significantly smaller than the nuclear genome, yet it holds a unique record of ancestry. Inherited through maternal lines, mitochondrial DNA forms a continuous thread that can illuminate migrations and population histories that may not be evident in nuclear sequences. It evolves at a different pace, gradually accumulating small variations that help track how groups of people spread across continents or became isolated in specific regions. Moreover, this genetic material contains essential instructions for energy production, making it vital for cellular function. When you examine mitochondrial DNA alongside nuclear DNA, you gain a dual perspective on human history and biological evolution. Together, they create a more comprehensive narrative than either could provide on its own. In nature, DNA can move between species in fascinating ways. Genetic material is more mobile than it initially seems. Certain microbes release tiny fragments of DNA into their environment, which other organisms can then absorb and incorporate into their own genomes. Additionally, some viruses serve as couriers, transporting bits of genetic information from one host to another throughout their life cycle. As you reflect on this interconnectedness, consider how it shapes the story of life on Earth. Would you like to explore more about these intricate processes? Imagine a world where even plants and animals can absorb genetic fragments from their parasitic partners or symbiotic microbes. This remarkable phenomena occurs under rare yet well-documented conditions. These transfers are intentional, not mere accidents. They happen through physical contact, environmental exposure, or interactions with tiny organisms that routinely exchange genetic material. When a fragment enters a new species, the receiving genome may interpret it as a new tool, a regulatory cue, or simply an additional stretch of sequence. Most fragments fade away without leaving a mark, but a select few can influence traits in unexpected ways, granting organisms abilities they never had before. Over long periods, this subtle movement of DNA forges surprising connections between distant branches of the tree of life. This process showcases one of evolution's unique approaches to exploring new possibilities, not just through gradual changes, but by sharing information across unexpected boundaries. Remarkably, some life forms possess far more genes than humans. While humans are often thought of as among the most genetically complex organisms, many species boast a significantly larger gene count. For instance, certain plants have experienced multiple rounds of genome duplication, resulting in vast collections of genetic instructions that enable them to respond swiftly to changes in weather, pests, and nutrient availability. As you reflect on this intricate web of life, consider the remarkable adaptability and interconnectedness of all living beings. Wouldn't it be fascinating to explore these connections further? Amphibians and fish exhibit remarkable genomic diversity, possessing vast genomes filled with extra copies of regulatory sequences, growth factors, and metabolic pathways. Even single-celled organisms can surprise you with their extensive gene sets, which enable them to adapt their lifestyles based on the chemical conditions surrounding them. This genetic richness doesn't necessarily equate to greater intelligence. Rather, it highlights varied survival strategies. Some species thrive on chemical diversity, while others depend on flexible metabolism or rapid environmental sensing. The range of gene counts across different life forms illustrates that evolution does not follow a single blueprint. It employs a unique combination of sequences tailored to help each organism thrive in its specific habitat, whether that results in a compact genome or one that spans millions of bases. Interestingly, DNA can endure in ice for astonishingly long periods. In frozen environments where temperatures remain low and biological activity nearly ceases, fragments of DNA can survive long after the organisms that created them have disappeared. As snow accumulates, it forms compact layers of ice, effectively sealing tiny pieces of genetic material away from heat, light, and oxygen. As you reflect on this intricate tapestry of life, consider how these adaptations shape our understanding of survival and evolution. Would you like to explore more about the mysteries of genetics? Or perhaps take a moment to rest and absorb this information? These protective conditions shield DNA from chemical reactions that typically cause damage. As a result, sequences can remain intact much longer than they would in other environments. Scientists have gathered material from ancient cores drilled deep into glaciers, as well as from the frozen remains of animals that once roamed landscapes that have now vanished. Each fragment holds clues about the organisms that shaped past ecosystems, including plants, microbes, and animals that have left no other trace. These preserved sequences allow researchers to reconstruct ancient communities, track climate changes, and understand how species adapted to environmental shifts over thousands of years. The ice serves as a molecular time capsule, safeguarding delicate information that lasts far beyond the lifespan of any creature. Within your genome, you carry genetic remnants from early life. Hidden in the long continuum of your DNA are faint echoes from biological worlds that existed before complex animals. These remnants originate from early viruses, ancient single-celled ancestors, and primitive molecular systems that once played crucial roles in the life of a young planet. Over vast stretches of time, many of these sequences have lost their original functions, yet they remain a testament to the intricate history of life on Earth. As you explore the depths of biological evolution, you discover how mutations accumulate over time. Active regions become quiet, and recognizable functions fade away. Yet even in this silence, fragments of early biological innovations remain. Some of these fragments resemble ancient enzymes that the first cells used to harness energy. Others appear to be remnants of early genetic elements that once moved freely among ancestral populations. By examining these molecular fossils, you gain insights into evolutionary episodes that lack any physical traces. They uncover the profound origins of essential biological processes, providing a glimpse into how life crafted its earliest biochemical tools before evolving into the diverse forms we witness today. Consider the remarkable nature of DNA. It can coil into nanostructures that are smaller than any machine humanity has ever created. The genetic material within a cell goes beyond simple spirals or loose strands. Under specific conditions, parts of this molecule can fold into incredibly tiny shapes, operating at scales that surpass our current engineering capabilities. These intricate shapes emerge when repeated sequences interact or when helper proteins guide the strand into compact forms. Some regions curl into tight rings, while others create short branching structures or tiny loops that stabilize delicate segments of the genome. As you reflect on this complexity, you can appreciate the beauty of life's architecture, a testament to the ingenuity of evolution. Would you like to delve deeper into this fascinating world, or perhaps take a moment to rest and ponder? Imagine a world shaped by intricate nanoscale architecture, guiding the movement of molecules and determining how they fit into confined spaces. Enzymes navigate specific regions with precision. The complexity of these structures makes direct observation a challenge, yet advanced imaging techniques unveil a hidden landscape within the genome, revealing subtle structures that shift as cells transition between states. DNA is more than just a carrier of instructions. It acts as a material capable of sophisticated mechanical organization. Some genes have origins that predate the Earth's first multicellular creatures, long before plants took root in soil or animals began to swim through oceans. Early microbes relied on a fundamental set of genetic instructions to carry out essential tasks. Remarkably, many of these ancient instructions persist in modern genomes, preserved because they continue to fulfill crucial roles. These ancient genes play vital roles in energy production, the replication of genetic material, and the management of key chemical reactions that sustain life. Their remarkable stability over billions of years suggests that nature discovered reliable solutions early in its journey and has continued to build upon them, even as life evolved into increasingly complex forms. When scientists examine these genes across diverse species, they uncover similarities that reach back to the earliest branches of the evolutionary tree. As you contemplate this intricate tapestry of life, consider the profound connections that link us to our ancient past. Would you like to explore more about this fascinating journey? Studying early life reveals the remarkable capabilities of the first cells, showcasing their resilience and the innovations that allowed them to thrive despite dramatic shifts in climate, chemistry, and planetary conditions. The speed at which DNA folds is one of the most rapid processes in biology. Within the nucleus, genetic material doesn't simply float in loose strands. It must quickly adopt orderly shapes right after it is produced. This folding occurs astonishingly fast, assisted by helper proteins that expertly pull, bend, and stabilize the DNA strand. Within seconds, long stretches of DNA are packaged into structures that not only protect delicate regions, but also ensure that different instructions remain accessible in the correct sequence. This rapid folding is crucial for maintaining cellular health. If the DNA were to remain unorganized, it would be vulnerable to damaging forces or become tangled, obstructing vital regions. Experiments capturing this process in real time demonstrate that DNA folding happens with remarkable coordination, almost as if the molecule remembers the shape it needs to take. This swift organization is essential, enabling cells to divide, repair damage, and respond to signals without hesitation. As you reflect on this intricate dance of life, consider how these microscopic processes shape the very essence of existence. The genome reveals itself as an active participant in both physical and informational realms. Some genes spring to life just once throughout an entire lifetime. Within the vast regions of a genome, there are instructions that activate for a fleeting moment, only to fall silent forever. These genes play crucial roles during early embryonic transitions, laying the groundwork for later development. They engage briefly, fulfill their purpose, and then retreat into silence for the organism's life. Other genes emerge during the creation of specialized tissues, lighting up just long enough to guide cells into their definitive identities. This brief activity demands remarkable precision. If these genes activate too early or too late, the entire developmental journey may stray off course. The presence of such genes illustrates that the genome functions not merely as a catalog of available tasks, but as a carefully timed sequence of events. Each instruction has a specific moment in which it must act, enabling complex bodies to evolve from simple beginnings. DNA is not just a passive vessel of information. It can instigate biochemical reactions simply by altering its shape. When a segment bends, twists, or relaxes, it transforms the chemical environment surrounding it. This dynamic interplay is essential for the intricate processes of life. A sudden shift reveals a hidden area that attracts specific enzymes or tightens regions, causing nearby reactions to slow down. Some shapes create small pockets where regulatory molecules fit perfectly, while others offer wide-open surfaces that encourage faster activity. These shape-driven influences enable the genome to guide biochemical processes without changing its sequence. Cells depend on these physical cues to determine which pathways to activate and how quickly to perform essential tasks. In this way, the structure of DNA directly participates in cellular chemistry. It sends subtle signals that help coordinate complex reactions in the crowded environment of a cell. Your immune system rewrites DNA to protect you. Specialized immune cells carry genetic regions that are designed to rearrange themselves. During their development, these cells cut and reconnect segments of their DNA, creating unique versions of defensive instructions. Each rearrangement allows the cell to recognize different molecular patterns on potential invaders. By generating countless combinations through this controlled rewriting, the immune system builds an extensive library of possible responses. When a new pathogen appears, at least one immune cell is likely to have a matching arrangement that can identify the threat. Your immune system possesses a remarkable ability to tackle unfamiliar microbes without the need to evolve new genes each time. This process is carefully regulated and confined to specific areas, ensuring that the rest of your genome remains stable. It represents one of the most advanced defense strategies found in nature, enabling you to thrive in environments filled with ever-changing organisms. Interestingly, some organisms store backup genetic instructions in unconventional locations. For many living systems, the nucleus is not the sole repository of crucial information. Certain species utilize innovative secondary storage sites that contain extra sets of instructions for emergencies, regeneration, or significant life changes. For instance, some single-celled organisms create tiny compartments that hold simplified versions of their genome, using these only during particular developmental phases. There are also insects that keep backup instructions within early embryonic tissues, allowing them to restart development when faced with stressful conditions. Plants, too, have their own strategies. They safeguard reserve sequences deep within dormant buds, which enables entire branches or even whole individuals to regenerate after enduring harsh winters or physical damage. In some unusual microbes, spare instructions are tucked away inside protective protein shells, shielded from toxins and radiation until conditions improve. As you explore these fascinating adaptations, consider how nature's ingenuity inspires resilience and survival in the face of challenges. Would you like to continue reflecting on these remarkable strategies, or take a moment to pause? Backups are not merely redundant copies of the same material. Instead, they often contain variations that provide flexibility in unpredictable environments. This hidden storage system adds resilience, illustrating how life creatively distributes information to ensure survival across ever-changing landscapes. Consider DNA, which encodes instructions for building proteins that can fold in trillions of different ways. When a protein forms inside a cell, it begins as a simple chain of building blocks arranged according to the DNA's instructions. As it emerges, it starts to shift, bend, and curl, ultimately settling into a unique three-dimensional structure. Each bend introduces a new shape, and with each shape comes a different function. The potential forms are staggering. Most shapes never materialize, as the chain naturally adheres to physical rules that guide it towards stable configurations. Yet, even within these constraints, the range of possible outcomes is vast. One sequence might fold into a structure that accelerates chemical reactions, while a slightly altered sequence could create a shape that transmits signals or transports molecules. This remarkable ability to generate a diverse array of tools from simple instructions is a key reason why life adapts so effectively. As you reflect on this, consider how intricately life weaves resilience and flexibility into its very fabric. Your genome serves as both a script and a generator for intricate machines, crafted from even the smallest structural changes. Within it lie hidden switches, capable of shaping behavior in profound ways. Scattered throughout your genome, these small regulatory sites influence when specific instructions are activated. They function like tiny switches, responding to chemical signals, developmental stages, or environmental conditions. Many of these switches don't directly create products. Instead, they adjust the activity of nearby regions. This adjustment influences how you respond to light, sound, memory formation, and stress. While behavior emerges from complex networks in your brain, these switches play a crucial role in shaping the underlying patterns that guide signal processing. Some switches modify the sensitivity of your neural pathways, while others dictate the speed at which messenger molecules appear or disappear. These regulatory regions vary from person to person, contributing to the subtle differences in temperament, attention, and emotional responses. They don't determine your destiny, but they help guide how your biological systems respond to experiences. Their presence illustrates that behavior is influenced by both your environment and deeply embedded control points that fine-tune the activity of your genetic instructions. So, as you reflect on your own experiences, consider how these hidden mechanisms might be shaping your responses in ways you have yet to fully understand. Some genes seem to emerge without any clear origin. Within the vast regions of the genome, researchers occasionally discover sequences that do not align with any known relatives in other species. These enigmatic genes don't resemble the ancient, conserved sequences that span across life forms, nor do they correspond to viral insertions or duplicated segments from nearby areas. They appear to arise suddenly in a lineage lacking a defined ancestry. Some of these genes may have evolved from stretches that initially served no specific purpose, gradually developing structure and function over time. Others might stem from rare occurrences where unusual chemical changes create new patterns that natural selection can later refine. These orphan genes can significantly influence traits unique to a species, ranging from specialized metabolic processes to distinctive reproductive strategies. Their existence poses a challenge to the notion that all genetic instructions must derive from ancient sequences. Instead, they illustrate that new genetic solutions can emerge independently, introducing fresh innovations into the ongoing narrative of evolution. Interestingly, DNA interacts with light and heat in wonderfully complex ways. While we often envision DNA as stable and inert, it responds to its environment in subtle and intricate patterns, especially when exposed to specific wavelengths of light. In certain regions, you notice subtle shifts in shape that lead to small structural adjustments. These adjustments can impact how easily a molecule unwinds. When heat is applied, some parts of the strand may loosen, while other areas tighten, Although these changes do not alter the genetic sequence, they significantly influence the genome's behavior. Cells depend on these minute reactions to sense temperature fluctuations, manage activity during seasonal transitions, and shield delicate regions from potential harm. In organisms facing dramatic environmental changes, these physical responses become vital for survival. They help regulate the speed of biochemical processes, determine when specific instructions are activated, and prevent stress from pushing the molecule past safe limits. This interplay illustrates that DNA is not just a chemical archive, it is a responsive material shaped by its environment, moment by moment. The genetic code is layered with redundancy. When cells interpret the genome, they adhere to a set of rules that convert sequences into the building blocks of proteins. These rules incorporate overlapping safeguards, minimizing the risk of harmful errors. Often, multiple combinations of symbols yield the same building block, meaning minor sequence changes typically have no significant impact. As you reflect on this intricate dance of life, consider how these processes quietly shape the world around you. Would you like to explore more about the wonders of genetics? Or perhaps take a moment to rest and absorb this information? In certain regions, small errors lead to gentle shifts rather than major disruptions. These built-in redundancies help maintain stability, even under stressful copying conditions. They allow evolution to explore new variations without risking catastrophic failure. By preserving meaning through alternative pathways, the genetic code ensures that essential functions continue, even as sequences drift over generations. This layered system reveals that life values both reliability and diversity. The genetic code isn't merely a simple translation chart. It's a carefully structured network that balances precision with flexibility. DNA may hold clues to forms of life we have yet to discover. Within the known diversity of genetic sequences, there are patterns that hint at biological possibilities not currently seen on Earth. Some repeated motifs appear to be remnants of ancient systems, with few living representatives, while others resemble structures predicted by theoretical models of alternative biochemistry. These unusual patterns suggest that early life may have experimented with strategies that either vanished or survived only in narrow environments. Take a moment to reflect on this intricate tapestry of life. What might these hidden clues reveal about our world and the possibilities that lie beyond? As scientists explore the genetic sequences of various species, they begin to uncover hints of pathways that defy current classifications. These intriguing clues spark curiosity about the possibility of life thriving in extreme environments on distant worlds, where the rules of chemistry might differ from what we know. While no direct evidence of extraterrestrial life has surfaced, the incredible variety within DNA showcases the flexibility of information storage. This diversity encourages the notion that life elsewhere could take on unique forms, all while relying on stable molecular systems to store and utilize information. Your own cells contain DNA that tells the story of ancient migrations across the globe. Many segments of your genome can be traced back to the long journeys undertaken by your ancestors. As populations ventured into new territories, they faced unfamiliar climates, foods, and diseases. Those who adapted successfully passed on genetic sequences that enabled their descendants to thrive in these new environments. Over generations, these adaptations accumulated in various groups, creating patterns that reflect the ancient paths traversed across continents, along coastlines, and over mountain ranges. Some sequences reveal expansions into the cold northern regions, while others indicate crossings through arid deserts or along fertile river valleys. Take a moment to reflect on these connections. The story of your DNA is a testament to resilience and adaptation, woven into the very fabric of life itself. If you're intrigued, consider continuing this journey of discovery, or take a moment to rest and let these thoughts settle. As you delve into the world of genetics, you realize that scientists are piecing together patterns that reveal migrations from a time long before written records existed. Each sequence, while not a complete story in itself, contributes to a larger map of human movement, shaped by chance, challenge, and exploration. These genetic footprints serve as quiet reminders of the diverse landscapes your ancestors traversed. Some organisms possess circular DNA, which acts as a second genome. Beyond the main chromosomes found in the nucleus, certain organisms maintain these extracircular strands that function independently. These loops hold crucial instructions for specialized tasks, ranging from metabolism to stress resistance. In bacteria, for instance, these circular DNA elements can carry genes that enable them to break down unusual chemicals or resist harmful substances. In the realms of plants and fungi, circular strands provide essential instructions that support unique life cycle stages. Even in animals, small circular fragments can emerge under specific conditions, often appearing in tissues that undergo rapid renewal. These loops have the remarkable ability to replicate autonomously, adjusting in number based on the needs of the cells. Their independence grants cells added flexibility, allowing them to activate alternative pathways without changing the core genome. As you reflect on this intricate web of life, you might find yourself wondering about the endless possibilities that arise from such remarkable genetic diversity. These circular elements serve as auxiliary libraries, providing essential tools that help organisms navigate unpredictable environments. DNA influences the lifespan of molecules within cells. Inside each cell, messenger molecules transport instructions from DNA to the structures responsible for building proteins. These messengers remain active for only a limited time before being broken down and replaced. The genome directly influences this timing. Specific patterns within the DNA sequence determine how stable these messenger molecules will be. This stability affects whether they linger long enough to produce significant amounts of a protein or fade quickly after a brief burst of activity. Cells fine-tune these patterns to regulate processes such as growth, stress responses, and metabolic rhythms. Some messengers degrade swiftly to prevent overproduction, while others remain stable to support sustained activity. This intricate control system ensures that the flow of information aligns with the organism's needs at any given moment. By managing the lifespan of these molecules, DNA maintains balance across countless biochemical pathways, coordinating timing with remarkable precision. Within your genome lie passages that have remained unchanged for eons. Across the vast expanse of evolutionary history, certain sections of the genome have stayed almost perfectly preserved. You notice that certain regions appear across many species that diverged from one another long ago. Despite their differences, their sequences remain surprisingly similar. This stability hints that even minor changes could disrupt essential survival processes. Many of these conserved regions play a crucial role in the early stages of development, directing how cells divide and organize. Others facilitate fundamental chemical reactions that all living organisms depend on. Because these regions have remained unchanged over vast stretches of time, they act as molecular anchors, connecting modern life to its ancient ancestors. When researchers examine these sequences, they can trace evolutionary relationships with remarkable accuracy. identifying shared patterns among mammals, birds, plants, and countless other lineages. Their existence reveals which genetic instructions have been so effective that nature has preserved them through shifting climates, mass extinctions, and the diversification of entire ecosystems. These conserved sequences provide a stable foundation beneath all the variation and novelty that evolution brings. Interestingly, some DNA replication methods are exclusive to exotic forms of life. While most familiar organisms utilize well-established molecular steps to copy their genetic material, certain unusual species employ dramatically different methods. This diversity in replication highlights the complexity and adaptability of life on Earth. Some viruses utilize unique mechanisms to replicate their genomes, employing a rolling process that cycles around circular strands. Other organisms take a different approach, breaking their chromosomes into numerous short fragments. They replicate each fragment individually before reassembling them into their original form. In certain deep-sea microbes, replication occurs in bursts, adapting to the extreme temperatures and chemical conditions of their environment. These diverse strategies reflect the specific conditions that shaped these organisms over time. They also illustrate how life can develop dependable copying systems from various origins. By examining these unconventional methods, scientists uncover valuable insights into how early life might have managed replication before the advent of modern systems. Furthermore, they discover new variations in genetic chemistry, enhancing our understanding of biological copying across the planet. DNA can even behave like tiny molecular springs. At a microscopic level, long strands of genetic material function like flexible wires, capable of storing and releasing mechanical tension. As these molecules bend around cellular proteins or twist during the processes of reading and copying, certain areas respond like small springs, stretching and compressing. These springs play a crucial role in distributing physical stress throughout the genome, preventing damage and guiding the movement of molecular motors that navigate along the strands. Isn't it fascinating how life adapts and innovates in such extraordinary ways? As you reflect on these processes, consider the remarkable resilience and creativity inherent in the biological world. Feel free to continue exploring this captivating topic, or take a moment to rest and absorb the information. The tension held within these tiny coils plays a vital role in how different regions of DNA open or close. Some springs relax to grant access, while others tighten to safeguard delicate segments. This mechanical action is essential for regulating genetic information. Without these spring-like behaviors, molecules would be more susceptible to breaks, tangles, and chemical instability. Their existence reveals how profoundly physical forces influence genetic behavior, even at scales too small for the eye to see. Among the diverse life forms on Earth, a select few are remarkable for their ability to repair damaged DNA far better than humans can. Certain bacteria withstand intense radiation by breaking their genomes into fragments and then meticulously stitching them back together. Some microscopic creatures endure complete dehydration that disrupts their DNA, yet they manage to repair it upon rehydration, returning to normal function without lasting effects. Additionally, certain plants exposed to high levels of natural chemicals or ultraviolet light employ robust repair pathways that quickly rectify damage. These extraordinary abilities stem from specialized enzymes, unique genome structures, and tightly coordinated repair networks that far exceed those found in humans. Take a moment to reflect on the resilience of life and the intricate mechanisms that allow it to thrive against the odds. Wouldn't you agree that there is much more to explore in this fascinating world of biological wonders? Exploring these remarkable organisms unveils strategies for maintaining stability in extreme conditions. It shows that life has developed various solutions for safeguarding genetic information. While humans depend on a specific set of tools, other species have evolved far more resilient systems, capable of thriving in environments that would challenge most forms of life. Within your genome lies a hidden landscape of regulatory elements. Beyond the well-known coding regions, there are extensive areas that function as control territories, shaping the behavior of entire groups of genes. And these landscapes are composed of clusters of small regulatory elements that work in harmony, determining when certain regions activate or remain dormant. Some of these elements regulate heartbeat rhythms, while others influence muscle development, immune readiness, or metabolic balance. The arrangement of these elements creates long-range interactions, allowing distant segments of the genome to coordinate shared tasks. Different tissues tap into various parts of these regulatory landscapes, which explains how the same genome can give rise to diverse cell types with unique functions. Identifying these regions can be challenging, as their effects often emerge only when large sections are altered. Yet, as researchers map their activity, they reveal sweeping patterns that illuminate how the genome orchestrates complex biological processes. As you reflect on this intricate interplay, consider how these insights might deepen your understanding of life's resilience. Would you like to continue exploring this fascinating topic, or perhaps take a moment to rest? These landscapes act like subtle guides, ensuring that instructions unfold with the right timing and intensity. Some genes seldom mutate because they are vital for life. Throughout the genome, there are regions so essential that they remain largely unchanged across many populations. Even minor alterations can disrupt critical processes, prompting natural selection to swiftly eliminate harmful changes. These essential genes often steer the basic functions of cell division, energy production, and structural organization. Since they must operate correctly in every cell, the margin for error is incredibly narrow. As generations pass, many individuals inherit identical or nearly identical versions of these sequences. This stability doesn't hinder evolution. Instead, it guarantees that the fundamental machinery of life stays consistent, allowing other parts of the genome to explore variations more freely. When scientists examine these rarely mutating genes across different species, they uncover striking similarities that reveal their deep evolutionary connections. They serve as the molecular backbone upon which more adaptable systems can innovate. DNA can even flip its orientation like a molecular mirror, with certain genomic regions capable of reversing their orientation. Feel free to pause and reflect on this remarkable journey of life and evolution. These segments revolve around a central point, flipping to run in the opposite direction. Specialized enzymes in cells manage this reversal, ensuring it occurs only when necessary. When the orientation changes, nearby instructions can dramatically alter their behavior. In some microbes, this mechanism allows them to switch surface features, helping them evade detection by predators or immune systems. In other species, these orientation flips control transitions between different life stages or metabolic states. This reversible system provides flexibility without changing the sequence itself. It acts like a rotational switch, toggling between distinct biological modes. This structural behavior reveals that genetic regulation goes beyond the mere order of symbols on the strand. It also involves the physical arrangement of the molecule in space. Some species possess genes that activate only in darkness. In environments where light cycles are crucial for survival, certain organisms have evolved genes that turn on exclusively when illumination fades. Deep-sea creatures rely on nocturnal instructions to manage their metabolism during the hours of darkness. Similarly, some plants utilize night-active genes to process energy reserves after sunset,